Good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET World. So yes, we have finished off Cell the Unit of Life in our previous session, and today we are going to exercise the EAPCET previous year questions based on the chapter Cell the Unit of Life, after which we are going to begin with cell cycle and cell division. So without much delay, let's get started with the first question you see. It is about A membrane bound organelles asked in the year 2023. Okay, A membrane bound organelles. If you see, ribosome and nucleolus both are non-membrane bound. Nucleolus, lysosome again is single membrane bound. Peroxisomes comes under microbodies which are single membrane bound. Lysosomes are again single membrane bound organelles. So here the right option goes with what? Option A. Second question you look into ma assertion reason type of question. Assertion reason type of question. Here you see ions and organic material concentration is higher in the cytoplasm than in the vacuole. True, right? Wrong, because the ions, and if you consider this as a cell, fine, right? If you consider this as a plant cell, we all know that in a plant cell, vacuole occupies 90% of the plant cell volume, okay, and the concentration inside the vacuole will be high and in cytoplasm will be low. That is the reason at the end or the last step of endomembrane system we have learnt that the molecules or ions are passed out against the concentration gradient, that is from low concentration to high concentration. Fine, right? So when this is the scenario, assertion will go wrong. Ions and organic material concentration is low in cytoplasm and it will be high in vacuole. Next, tonoplast facilitates the transport of ions and other materials against the concentration into the vacuole. Reason is true. So here A is false and R is true. We have to go with option D. Next, match the following type of question based on the shapes of cells. Here we go with shapes of cells. White blood cells, we know white blood cells are amoeboid. White blood cells are amoeboid. Next, mesophyll cells. Mesophyll cells are round and oval. Red blood cells round and biconcave. Tracheate cells are elongated cells with tapering ends. So here A is 3, B will be 4, excuse, C is 2 and D will be 1. So it is A3, B4, C is 2 and D will be 1. Option C. Okay. Next. Here we go with functions which are not related to cytoskeleton. Before answering this question, you have to recall or memorize like what all are included in the cytoskeleton. Cytoskeleton includes proteinaceous network, which includes what ma? Microtubules, microfilaments, and intermediary filaments. Microtubules, microfilaments and intermediary filaments. So here if you see functions which are not related to here is like, yes, of course, cytoskeleton, all the proteinaceous network will give the mechanical support, true, and they help in cell motility, true, intracellular transport, true, extracellular transport also will go correct, but not cytokinesis, okay, but not cytokinesis. So here 1, 2 and 4 are the functions which are related to cytoskeleton except 3. So here the option is C. Next, assertion reason type of question. Here we go. Suitable stage to observe chromosome in metaphase. Suitable stage to observe chromosomes is metaphase. True, yes. In metaphase we can observe the chromosome structure in detail. In metaphase, chromosomes are scattered in the cytoplasm. Reason is wrong, because in metaphase stage chromosomes are aligned to equatorial plate. Fine, right? So here assertion is true and reason is false. We will go with option C. Next question you look into, ma. Yeah. Which of the following structure is found in all prokaryotic cells? In all prokaryotic cells, you will see only non-membrane bound organelles. Right away in this options, if you consider, the non-membrane bound organelle is ribosome. Okay. Apart from that, if you see other organelles, mitochondria is of course double membrane bound. Fine, right? Golgi body single membrane bound, lysosome single membrane bound. So here the option is C. Okay. Next, look at this, ma. Again, match the following type of question with regards to discoveries asked in EAPCET 2022. Look into it and answer carefully. Yeah. Here we go. First is what? Leeuwenhoek. Okay. Leeuwenhoek is the first scientist who identified the living cells. Okay. He observed different kinds of cells. First to observe what? Living cells. So A is option 4. Next, Robert Brown is the first to identify nucleus in the root cells of orchid. So this is 5. Next, Theodore Schwann. He is a British zoologist, but then he proposed the point about limiting boundaries both in the plant cell and animal cell. He is the scientist who said the limiting boundary of plant cell is cell wall. So this option is 2. Matthieu Schleiden. Okay. Schleiden has observed both the plant cells. Schleiden has only worked out on plant cells, and he said that plant body is composed of cells. Observe different kinds of cells forming tissues in plants. Here it is option 1. And Rudolf Virchow is the only scientist who has proposed omnis cellula e cellulae. Omnis cellula e cellulae. Fine, right? So Rudolf Virchow has proposed this, which is option 3. Fine, right? So A is 4. Just a second. Yeah. A is 4. B 5. C is 2. D option 1. And E is 3. So what is the right answer? Option C is the right answer over here. Clear? Any doubt anywhere, ma? Yeah. Next question you see. Assertion reason type of question again. Growth in the primary wall of young plant cell. This gradually diminishes as cell matures. Yes, right? Growth gradually. Growth of the primary wall of the young plant. Primary wall of the young plant. This gradually diminishes as cell matures because secondary wall layers will be decreasing the space. This will be increasing the space for cytoplasm and decreases the space for the primary cell wall. Thereby, primary cell wall layer gets disintegrated as the cell wall, as the cell matures, and as the secondary cell wall layers get increased. So here growth in the primary wall of the young plant cell, this will gradually diminish as the cell mature. This is true. Next, secondary wall is formed on the outer side of the cell. No, secondary wall is the last layer that is formed, and it is formed towards the inner side of the cell, which is close to the cytoplasm. So assertion is true and reason is false here, which is option C. Next, assertion reason type of question. If you have observed all the set of questions, beta, most of the questions are directly from your text lines, directly from your academy book lines. Okay, and mostly you will see assertion reason type, match the following, and statement based questions only from this chapter. Here we go with assertion reason type. You see, assertion, cytoskeleton in a eukaryotic cell is made up of network of filamentous lipids and polysaccharides. Cytoskeleton is composed of microfilaments, intermediary filaments and microtubules. True, right? Here you see reason is correct, but once again you look at the assertion. Cytoskeleton in a eukaryotic cell, this is made up of what? Network of filaments of polysaccharides and lipids. No, they are made up of network of filamentous proteins, not lipids and polysaccharides. So here the right answer will be assertion is wrong and reason is true. Okay, option D. Next question, again match the following type, just go through it, ma. Just go through it. Yeah, here we go. Tonoplast is the outer boundary for what? Vacuole. Tonoplast is the membrane which is covering the vacuole. And stroma, which gives the enzymes for connection. Just a second. Plasmodesmata. Plasmodesmata which is present. Plasmodesmata is the only living component that is present in the cell wall which connects to adjacent cells. So D is option 3. And CUC, cisternae of Golgi, helps in glycosylation and glycosylation of what? Proteins, modification of proteins. And next, if you observe, stroma. Stroma contains enzymes for anabolism. Okay, because here it prepares inside stroma. What happens, ma? Glucose synthesis happens. Stroma, which is known as what? Matrix of chloroplast. Stroma, which is addressed as matrix of chloroplast. Fine, right? Stroma, which is the matrix of the chloroplast. Inside chloroplast, glucose formation takes place. So stroma consists of the enzymes which are required for anabolism. And here we go with tonoplast. Tonoplast, which is the outer layer of vacuole, it will help in the osmoregulation between cytoplasm and vacuole in a plant cell. So which is option 4. A is 4, B is 5, C is 1 and D is 3. Here the right answer is option A. A is the right answer. Next question you look into. Here we go. Assertion reason type again. Assertion, lysosomes are capable of digesting proteins, lipids and nucleic acids. True. Lysosomes are rich in hydrolytic enzymes. Correct. Okay. So assertion and reason both are correct here, and reason is the correct explanation of assertion, because lysosomes are capable of digesting all this because they are rich in hydrolytic enzymes. This is how reason is explaining the assertion over here. So we can go with option A. Okay, A and R are true and R is the correct explanation of A. Here we go with match the following type of question with multiple columns. Okay, look into that carefully and address the question. Look into it carefully and address the question. See, first you see circular DNA. Okay, circular DNA is present in what? Prokaryotes. Prokaryotes, they will not have any nuclear membrane, no? Yeah, so A goes with option 4. And here you see, there is no role in development of cell plate. Yeah, this has what? Plasmid DNA in a bacterial cell. Circular double-stranded DNA which doesn't have any nuclear membrane, and this you will see in a plasmid DNA. So A, 4 and 3. Okay. 4 and 3. Next you see fimbriae, which are bristle like structures that are sprouting out of the cell. B1. Fine, right? Next you see here, this helps in attachment of the bacterium to the host tissue. So here B1 and 1. Next 3 you see beta, middle lamella, which is the outermost wall layer. Once you recall, middle lamella is the first formed wall layer. Inner to the middle lamella, what is formed? Primary cell wall is formed. Next to the primary cell wall, you will have 2 to 3 layers of secondary cell walls. Okay. So first it is middle lamella, followed by primary cell wall, followed by 2 to 3 layers of secondary cell wall. As such, secondary cell wall will be closer to the cytoplasm and middle lamella will be the outermost wall layer of the cell. So here you see C is middle lamella, which is the outermost layer of the cell wall, and this helps, this is seen in what? Plant cells, right? So this will develop from what? Cell plate. Okay. So middle lamella, which is the outermost wall layer, and this develops from where? Cell plate. Cell plate only matures to middle lamella, which is the first formed wall layer in the plant cells. C, 2 and 4. Okay. C, 2 and 4, which is option 2. Okay. Next you see D, that is mesocarp and endocarp fruit. Mesocarp and endocarp fruit fused. This you see, the best example is Macaulay, which belongs to which family? Solanaceae family. So D is 3 and 2. Okay. What will be the right option here? Option B. Clear, right? Everybody understood this question? If you are having any doubts, you can drop a message in the chat box, ma'am. Anybody, any doubts? Next we go. Yeah. Here you see statement based questions. Choose correct statements among the following. H1 histone molecules lies outside the nucleosome and this will seal 2 turns of DNA. Yes, A is correct. Next B you see. Negatively charged DNA and positively charged histones will allow packaging of DNA. True? Yes, this will allow what? Packaging of DNA. Each nucleosome has 6 histone molecules and 145 nucleotide pairs. Wrong, because each nucleosome will have 8 histone molecules, which is histone octamer. Right, this is what? Histone octamer. And how many nucleotide bases it will have? 200 base pairs of nucleotides. So this is wrong. DNA between 2 successive nucleosomes is called as linker DNA, which has H1 histone proteins. True. So here A, B and D are correct. C is wrong. Option D. Okay. Next assertion reason type of question you see. In vacuole, the concentration of ions and other materials are less than the cytoplasm. Wrong. See, this question is repeated. Find right. And in tonoplast facilitates the transport of ions and other material against the concentration gradient. True. So A is wrong and R is true. A is false and R is true, which is option D. Repeated question mark. Next, fill in the blank type of questions. Very important. Paragraph based questions. So here we go with chromosome structures. Okay. Chromosome shapes, which is clearly visible in which phase of cell division? Anaphase of cell division, right? So here you see each chromosome has a dash or dash on the sides. What do they have on sides? On sides they do have satellite DNA. Each chromosome has on the sides, just we will keep a pause over here. Disc shaped structure is called as what? Kinetochore. Disc shaped structure around the primary constriction is addressed as what ma? Kinetochore. Fine, right. Next, the dash chromosome has 2 equal arms. Which type of chromosome will have 2 equal arms? Metacentric chromosome. And dash chromosome has one short arm, one shorter and one long arm. Short and one long arm is submetacentric chromosome, that is E. Okay, one short arm and one longer arm. And dash chromosome has no second arm at all. No second arm, you will see it in telocentric chromosome, where one arm of the chromatid is negligible totally. Right or wrong? Yeah, here the option is B. Fine, right ma. Next, the secondary constriction develops a small fragment called as secondary constriction will develop a small fragment that is addressed as what ma? Satellite. That is F. Each chromosome has one primary constriction. That primary constriction is also addressed as centromere. So, here it is DC followed by kinetochore, that is G followed by A and then E followed by B and F. So, here the right answer is option B. Any doubts anywhere? Clear with this? Any doubts anywhere ma? Just look into it and answer. If you are having any doubt, you can ask me. Fine, right, because paragraph based questions, many students will make mistakes over here only. Okay, these are present only in the EAPCET exam. This type of questions you will not see in the NEET. Okay, so whoever are having doubts, you can ask me. Next, what are the common characters of both prokaryotic and eukaryotic cells? Okay, we have clearly learnt now the differences between prokaryotes and eukaryotes and the common features between prokaryote and eukaryote. Of course, cytoplasm is present both in prokaryote and eukaryote because this is the main arena of all cellular activities. Plasma membrane, of course, it is present in a prokaryotic and eukaryotic cell. Mitochondria is absent. Next, cellulosic cell wall is absent and meiotic cell division is absent in prokaryotes because in prokaryotes amitosis takes place. Fine, right? Amitosis occurs in prokaryotes. So, here A and B both are true. Option B. Clear, right ma? Next, you see. Choose the correct statements. Polar molecules cannot pass through non-polar lipid layer without carrier proteins. Okay. So, polar molecules, they cannot pass through non-polar lipid bilayer. True, right? They cannot pass through non-polar lipid bilayer without carrier proteins. Yes, true, because polar molecules are transferred by what? Facilitated diffusion. So, A is true. Next, plant cell vacuole plays role in osmoregulation. True. That is because of what? Tonoplast. This point has raised twice in your EAPCET exam. This is what? Tonoplast. Next, you see, Golgi apparatus is the site for synthesis of proteins and glycolipids. It is not for the synthesis of proteins, it is for the synthesis of glycoproteins and glycolipids. Protein synthesis happens from where ma? RER, that is rough endoplasmic reticulum. This is by glycoproteins and glycolipids. You to identify correct set of statements or incorrect set of statements. A, B and T, these are the correct set of statements. Option B is the right answer. Next question, you see. Yeah, which of the following synthesizes the cell wall? Okay, which of the following synthesizes the cell wall? Cell wall is synthesized from Golgi bodies because because Golgi bodies will synthesize the first material which is rich in calcium pectate and magnesium pectate, that is phragmoplast. Phragmoplast is synthesized from Golgi bodies. Phragmoplast will develop to cell plate. cell plate matures to middle lamella. Middle lamella. Fine, right? So this is how you have to project it. Which of the following synthesizes the cell wall? Right, ma? From Golgi bodies, phragmoplast is synthesized. Phragmoplast will mature to cell plate will mature to middle lamella. Next question you look into, arrange the following in ascending order of their numerical number. So look at each question carefully, each statement carefully. Total number of peripheral fibrils in a centrosome. Okay, centrosome. In a centrosome, first you recall, in a centrosome you have two perpendicularly arranged centrioles. Yes or no, ma? You have two perpendicularly arranged centrioles. Two centrioles are present in one centrosome. If you take the transfer section of one centriole, you have 9 radial spokes in each centriole, 9 radial spokes. Each radial spoke will have 3 peripheral microfibrils. So 9 into 3 it is 18, right? 8 peripheral fibrils are present for each centriole. As such, you have 2 centrioles in a centrosome. 2 centrioles means here 18 plus 18, you will have how much, ma? 32 peripheral microfibrils. So it is 32 peripheral microfibrils are present. Option A. Next, B you see, total number of microtubules in an axoneme of cilium. Cilia and flagella will have what type of microtubular arrangement? They will be having 9 plus 2 microtubular arrangement, wherein they have 9 radial spokes. Each radial spoke will have 2 peripheral microtubules and they will have 2 central microtubules which are enclosed by a central sheath. As such, if you see here, 9 into 2 how much, beta? 18 plus 2, you will have 20 microtubules on total. Okay, you will be having how many microtubules? 20 microtubules on total. Next, number of arms in a telocentric chromosome. In a telocentric chromosome, you will have only one arm of the chromatid, whereas the other arm of the chromatid is negligible. Next, number of nucleotides in a nucleosome. How many nucleotides will be there in a nucleosome? See, first thing, a nucleosome consists of 200 base pairs. Yes or no? 200 base pairs. Means how many nucleotides are present? 400 nucleotides are present. Fine, right? Because each nitrogen base is for one nucleotide. 200 base pairs are present. So, how many nucleotides on total, ma? 400 nucleotides. Now, you arrange it based on the numerical order. First, it will be what? Telocentric chromosome, which is option C. Yes or no? Followed by what? Followed by B. Okay. So, here total number of microtubules of cilium, it is 9 plus 2, and here if you consider in a single centriole, of course centrosome will have 2 centrioles, but if you consider single centriole, it will be 18. In that case you will have C followed by option A followed by option B and then D. This will be the answer. But then in a complete centrosome, you have 2 centrioles peripheral microtubules. If you see 32 peripheral microtubules you will have. In that case, the option will be C followed by B followed by option A and then this is followed by option D. Clear, right? This will be the right set of answer in the ascending order. Clear, right? C, B, A, D. Done. Next you see, ma. 21st question. Again statement based only. In the paragraph type he has asked. Fill up the blanks with suitable words. Okay. So here we go with this. Yeah. Schleiden observed that all plants are composed of different kinds of cells. Schwann studied animal cells and he reported that plants have cell wall, right? It is Schwann who has reported plants will have cell wall. So, here it is option D, fine, right. Next, and all the cells will have plasma membrane, fine, right. So, D will be here first and followed by A, just a second, first it is D followed by, all cells will have what? Plasma membrane is present in all the cells, and here there is no blank in between, right, but Schleiden is a botanist, here we need to include F. Schleiden is a botanist, fine, right. Observe that all plants are composed of different kinds of cells, and then here few blanks have missed in the question, we will exercise it later, because in the beginning of the question it is missed, but then from this paragraph the content that you have to learn is, Schleiden is a German botanist, both Schleiden and Schwann has proposed cell theory that we know, and he has observed only plant cells. Schwann has given out the limiting boundaries both for the plant cell and animal cell, and it is Rudolf Virchow who has proposed omnicellula e-cellulae concept, that is all new cells arise from pre-existing cells. Here we go with the next question of nucleosome model. Yeah, nucleosome model, look into it, ma'am. Okay, here we go. A typical nucleosome bead is made up of, one bead in the nucleosome is made up of what? 8 histone molecules, which is a histone octamer, and 200 base pairs, which is option B. Okay, next you see, match the following type of question. Here we go. Amyloplast consists of starch or carbohydrates. Elioplast consists of oils or fats. Aleuroplast consists of proteins. Leucoplast consists of what? Leucoplasts are colorless plastids which will store the nutrients, which is option 1. Okay, clear? Next. Yeah. Which of the following characters are true for ribosomes? So, here the question is about ribosome characteristics. Look at it carefully. Yeah. Composed of RNA and proteins, and they are non-membrane bound. Yes, ribosomes are nothing but ribonucleoproteins. Next, membrane transports are membrane transports a number of substances. Wrong, because it does not have any membrane. Membrane bound vesicles in hydrolytic enzymes, no, they will help in protein synthesis. Of course, ribosomes primary function is what? Protein synthesis only. So, here the right answers are 1 and 4. Option A will be the right answer. Next, assertion reason type you see, ma, assertion reason type of question. Assertion, fluid mosaic model explains quasi-fluid nature of lipid bilayer allowing lateral movement of proteins. Yes, this is true. Next, in young plant cells, primary wall grows and later growth diminishes as secondary wall is formed in the inner side. True. So, both A and R are correct over here, but nowhere there is a connection between the fluid mosaic model and cell wall formation. There is no point of proper explanation here, but then A and R are true, but R is not correct explanation of A. Okay. So, with this we have finished out the complete unit of cell, the unit of life with EAPCET questionnaire practice. Now, we will move on to cell cycle and cell division. Before getting into the chapter, I will give you a brief note on this chapter, like from where we can exercise most set of questions. Here we go. First, you have to know the difference between cell cycle and cell division. Cell cycle is nothing but what, ma? cell cycle is complete sequence of events which includes duplication of a genome, synthesis of new cellular organelles, and finally formation of two daughter cells. This complete sequential events comes under cell cycle. Okay, what does it include, beta? It includes duplication of genome, synthesis of new cell organelles, and synthesis or duplication, or you can also consider formation of daughter cells. Formation of daughter cells. This complete sequence of events at the end, formation of new daughter cells, this complete sequence of events is addressed as what? Cell cycle. But cell division is the actual phase of M phase or division phase. Cell division consists of what? Actual division phase. This consists of actual division phase. Cell division consists of actual division phase where the cell divides, nucleus divides. This includes both karyokinesis and cytokinesis. This includes both karyokinesis and cytokinesis. Right, ma? Now, cell cycle, as it involves sequence of events, it has two phases. First is what? Interphase. Interphase. Interphase is also addressed as biosynthetic phase, or it is also addressed as resting phase. Fine, right? Or it is also addressed as preparatory phase. So here if you see, why those many synonyms for interphase? In interphase actually cell doesn't divide, cell prepares for a division, and this itself occupies 90% of cell cycle time. Interphase itself occupies how much time, beta? 90% of cell cycle time is occupied for preparation only. Even if you consider your NEET examination, for whole year you will be preparing, might be you have taken a long term, or if you are in 12th standard to 11th, 12th, 2 years you will be in the NEET preparation or EAPCET preparation. But then your exam tenure will be just 3 hours. So, maximum time is consumed for preparation and less time is for execution. Best preparation without any errors will give you proper execution. In the same way, without any errors, the cell completely prepares itself for division, wherein for M phase, that is for karyokinesis and cytokinesis, it takes only 10% of time, 10% of time for cell division. Okay, M phase only takes 10% of time for cell division. So here you see interphase, biosynthetic phase, resting phase, because there is no actual cell division, and it is also called as preparatory phase. Biosynthetic phase because it synthesizes so many molecules here in this division. So here interphase has 3 subphases. One is G1 phase, followed by S phase, followed by G2 phase. And M phase can be of 3 types, ma. N phase can be of 3 types. One is amitosis, second is mitosis, third is meiosis. I will explain you all the 3 in brief. Listen, but here the flow chart is clear, right? With respect to your exam, you will get numerical questions addressing meiocyte, meiospores, mitospores, mitocyte in connection with sexual reproduction of flowering plants might be, ploidy from plant kingdom might be. You will get the questions in connections with numerical based. Okay. So, here we go with amitosis, mitosis and meiosis. So, as we have discussed, cell cycle will have what ma? Interphase and M phase. Here you can see interphase. It has G1, S and G2. In between G1 phase and S phase, there will be one major checkpoint which is addressed as G1S checkpoint. Okay. Here you will have one major checkpoint that is addressed as what? G1S checkpoint, which is a major checkpoint. This checkpoint will have G1 cyclins and CDC2 kinase. Fine, right? And in between from G2 to M also you will have one checkpoint that is addressed as G2M checkpoint. G2M checkpoint. Fine, right? This has G2 cyclins, G2 cyclins and CDC2 kinase. What is the role of these checkpoints? These checkpoints will check for the proper genome duplication and proper cell organelles duplication and proper synthesis of the organelles without any errors. It will help in the pass of the next phase. It will help the cell to pass on to the next phase, right? So, G1S checkpoint is addressed as major checkpoint and G2M is addressed as minor checkpoint. Fine, right ma? So, this is about G1, S and G2 phase. Here we go with first amitosis, types of cell division. Here I hope interphase, checkpoints and M phase is clear to you, right? Now, first we will move on to amitosis. First is amitosis. This type of cell division is mostly observed in unicellular organisms. Unicellular organisms wherein there will not be any requirement of spindle apparatus. There is no requirement of spindle apparatus or spindle fibers. Okay. The cell duplicates as such, the genome duplicates as such, okay, without any involvement of spindle fiber apparatus. That is the reason it is addressed as amitosis. Commonly it is well noted as what? Binary fission. Binary fission. Fine, clear till here? And here you have to make out one more point, that is M phase, as we have discussed, be it mitosis or meiosis, it will be having out what ma? Karyokinesis, which is nuclear movement or nuclear division, followed by cytokinesis. Okay, karyokinesis followed by cytokinesis. Karyokinesis, here if you see, we have prophase, 4 substages in karyokinesis: prophase, metaphase, anaphase and telophase. Cytokinesis, if you see, that is cell division, it will be different both for plant cell and animal cell. Fine, clear? You are clear on this note, I hope. Now we will move on to first is G1 phase. If you see G1 phase, in G1 phase what happens is G1 phase of interphase. Today we will discuss all the phases of interphase, and in our next class we will move on to M phase beta. Both mitosis, meiosis, including numerical questions we will solve. So, here if you see in G1 phase, most of the cell organelle duplication happens in G1 phase. As most of the cell organelle duplication or cellular constituents are synthesized in G1 phase, the cell size will increase, okay, and cell is metabolically active in G1 phase, okay. Cell is metabolically active, here cell size increases and most of the organelle duplication, most of cell organelle duplication occurs in G1 phase. Clear, right? Be clear on this note. And most of the cellular constituents are synthesized in G1 phase only. Cellular constituents synthesized in G1 phase. Clear? Now we will move on to S phase or synthesis phase. In S phase or synthesis phase, two events will happen. One is inside the nucleus. One is inside the nucleus. Second is inside the cytoplasm. So, inside nucleus what is happening you see. First is inside nucleus. Inside nucleus DNA duplication happens. DNA duplication occurs without any chromosomal duplication. How does this happen? Chromosomal duplication. First you keep this in your mind that you observe the chromosome structure in the metaphase clearly. Till then the chromosome will be in the form of chromatin. But to make you clear on the note that chromosome duplication doesn't occur in the S phase of interphase, they have used the term chromosome in your text. But then chromosome structure in actual sense you will see it in metaphase only. This is hypothetical. But to make it clear for you, I will give you one small diagram. Let us suppose this is one chromosome, right? Two chromosomes are present before the S phase in G1 phase. Now after duplication also it will be 2 itself. Fine, right? There will be no chromosomal duplication, 2 chromosomes itself. Only DNA or the genetic material will get duplicated. This is inside the nucleus. Second event that happens is inside the cytoplasm. Second is inside the cytoplasm. Fine, right? So inside cytoplasm what happens, beta? Inside cytoplasm centriole duplication occurs. Centriole duplication occurs. Fine, right? Centriole duplication in the cytoplasm. These centrioles which are duplicated in the S phase will help in the spindle apparatus formation during anaphase of the cell division. Clear, right? So if you consider this as a cell, okay, this as a cell, 2 centrioles are formed. Priorly it will be single centriole or centrosome you consider, and after S phase 2 will be formed as such. Fine, right? Next is what? G2 phase. In G2 phase the cell organelle duplication will happen. There is a popular statement which is asked multiple times in your NEET and EAPCET that semi-autonomous or self-duplicating organelles will replicate or duplicate in G2 phase. Semi-autonomous or semi-autonomous or self-replicating organelles. What are they? Mitochondria and chloroplast. They will duplicate in which phase? G2 phase. Fine, right? Along with these two, Golgi bodies are also duplicated in G2 phase. Golgi bodies. And the protein synthesis like tubulin proteins which are helpful in the spindle apparatus formation are synthesized in G1 phase. Okay, so I hope now you have got a clear idea on G1 phase, S phase and G2 phase. This is about the complete cells which are dividing very often or very frequent. Now let us talk about those cells which will not divide very often. Okay, let us discuss about those cells which will not divide very often. Those cells like brain cells or heart cells. Okay, brain cells or heart cells which will not divide very often. Yeah, they will enter into G0 phase. So, G1 phase tenure is variable based on the cell. The G1 phase time will be variable. To the cells which will not divide very often, like heart cells or nerve cells, they will exit the G1 phase, and whenever they are resuming back their cell division, they will get back to G1 phase again. So, these cells will exit the G1 phase and they will enter G0 phase. G0 phase is also addressed as quiescent stage. Quiescent stage. Cells in this stage are metabolically active. From G0 phase, it will again move on to G1 phase only, from G1 to G0 and again to G1. Clear, right? So, this is the complete picture about the interphase. In our next class, we will move on to mitosis and meiosis in detail, where we will finish off all karyokinesis, cytokinesis, and we will also have a numerical approach based questions. Okay. So, till then, practice till here. Look into all the EAPCET questions. If you are having any doubts, you can drop a message in the chat box. I will be addressing all your doubts. Okay. So, till then, keep revise, be updated with this concept, and in our next class we will look into the mitosis and meiosis concept. Thank you.